Hello, Life Science. Hope you're doing well. Today, we're going to continue Chapter 6, Role and Components of the Immune System, with the last document, Document 7, Antigen Recognition by T lymphocytes. Let's first do a quick review about the antigen recognition by B lymphocytes. So you know that B lymphocytes recognize directly the antigen. How? This occurs through BCR or antibodies. So you have in the figure below a scheme that represents the structure of antibodies. Antibodies are made up of 4 polypeptide chains, 2 heavy chains, and 2 light chains. The light and the heavy chain are bound together by disulfide bridge. Also, the 2 heavy chains bound to each other by 2 disulfide bridges. Every antibody consists of a constant region and of 2 variable regions. The variable region contains the antigen binding site. An important note to be said here, which is antibodies found on the surface of lymphocyte B or even the soluble antibodies, they bind only to extracellular antigens, which are the antigens that are outside the cells and not inside the cells. And that's why we say that B lymphocytes recognize directly the antigen before it enters and infects any cell. We can start now with document 7, antigen recognition by T lymphocytes. The main objectives of this document, first, to explain the recognition of the antigen by T lymphocytes. Second, to describe the immune response against an antigen. I'm going now to explain briefly the steps of the immune response. These steps are going to be discussed in details in the next chapter 7. But going through these steps actually will help us understand better how do T lymphocytes recognize the antigen. So what are these steps? When I talk about immune response, it's the response of the immune system. Of course, a response for what? It's a response for an antigen that has entered the body. This means that there should be an infection. So in case of infection, the non-self enters the body, so the immune system should be activated. How does this happen? It occurs actually in 2 steps. A first step, which is triggered within few hours after the infection, and it is called a non-specific immune response. It's immediate response. The effector, that is the cell that is going really to eliminate the antigen, is the macrophage. How this is going to happen through phagocytosis, through digestion of the non-self. In case the non-specific immune response fails to eliminate the antigen, so if the macrophage fails to eliminate the non-self, what happens then? A second immune response is going to be triggered, and we call it specific immune response, which is the second line of defense. It's a delayed response because it takes time to be triggered and slow. The main effectors of this immune response are the lymphocytes B and T. It is slow, yes, but it is actually more efficient than the non-specific immune response. What do you think? Why did we call the first line of defense a non-specific immune response, while the second one a specific immune response? Well, non-specific immune response because it doesn't depend on the identity of the intruder, which means that the macrophage can digest any kind of non-self, whether it's a virus or a bacterium. However, the specific immune response, it depends on the identity of intruders, which means that LB, for example, fight a specific type of non-self, while LT, they fight other kinds of non-self. So the immune response here, it depends really on who is the non-self. Okay, so we said that if the macrophage fails to eliminate the antigen in the non-specific immune response, then there will be induction of a second line of defense, which is the specific immune response. Actually, I'm not going to go to talk about the non-specific immune response for the moment, but I care for the role of the macrophage in the specific immune response. The macrophage is going to be an antigen presenting cell, or APC. What does this mean? The macrophage, if it is unable to eliminate the antigen by itself, it is going actually to synthesize peptides, and then these antigenic peptides will be displayed on the surface of the macrophage. Where exactly? The macrophage is an immune cell that possesses, in addition to HLA class 1, it possesses HLA class 2. So the antigenic peptide, as you see in the figure, is going to be displayed on the HLA class 2. Okay, so how is this macrophage going to be an APC? How is it going to present the antigen? Actually, it is going to present a sample of the antigen, which is the antigenic peptide. But to who is this presentation going to happen? It is going to happen to T4 lymphocyte or T helper. T4 lymphocyte actually has to do a double recognition. A recognition of HLA class 2 and a recognition of the non-self peptide. Hence, when T4 lymphocytes recognizes these two elements, we can say T4 lymphocyte is activated. And it is the main lymphocyte in the immune response. So T4 lymphocyte recognizes the antigen carried by HLA class 2 on the surface of macrophage, which is the APC. So this is the role of macrophage in the specific immune response, and this is one way of recognition of the antigen by T lymphocyte, which is when T4 recognizes indirectly the antigen. How? By making a recognition of the peptide, antigenic peptide of course, and a recognition of HLA class 2. See another way T lymphocytes recognize the antigen. This is a cell infected by the virus. So the virus, which is an intracellular antigen, which means an antigen that enters inside the cell, the cell displays the viral peptide. After synthesizing, of course, these peptides, the cell is going to present the viral peptide on HLA class 1. Why class 1? Because the cell that could be an intestinal cell, a pulmonary cell, a hepatic cell, it doesn't possess HLA class 2. So it's only HLA class 1 present on its surface. That's why the antigenic peptide will be displayed on HLA class 1. Okay, which type of T lymphocytes are activated here? As you see in the figure, it is T8 lymphocyte. Why T8 lymphocyte? Because T8 lymphocyte, mature T8 lymphocyte, are characterized by their ability of making a double recognition. A recognition of the self HLA class 1 and a recognition of the non-self peptide. So the T8 lymphocyte here, okay, it can recognize the antigen, which is a sample of the antigen, the antigenic peptide, while be carried by HLA class 1 on the surface of self-infected cells. It's not only on the surface of self-infected cells, it also could be a tumor cell that presents on HLA class 1 and on self-peptide, or it could be also a cell of a grafted organ. Okay, I want you please to open your book on page 131, where you have this concept map, to do a summary of what we have seen in chapter 6. Okay, so you can see in the primary lymphoid organs, which are the bone marrow and the thymus, LB and LT are produced in the bone marrow. LB mature also in the bone marrow, while LT are still immature. They move into the thymus where they become mature. Okay? Now, these mature LB and mature LT, the monocyte and the granulocyte, they move into the blood vessels. Now, in case of infection, first of all, the granulocyte is a cell that is actually able to digest the non-self by phagocytosis. So you see in the figure, the granulocyte can move in the tissue, into the tissues where it undergoes phagocytosis of the non-self, of the antigen. The monocyte also, just like the granulocyte, is a cell that is able to do phagocytosis, but it is modified into a bigger cell called macrophage, and at the level of the tissues, the macrophage can digest the antigen or the non-self. And this is what we called in the previous slide the non-specific immune response. The macrophage and the granulocyte, they don't care about the identity of the intruder. All intruders can be killed alike in the same way by phagocytosis. Okay. Second, in case the pathogen is not eliminated, so in case the non-specific immune response failed in the elimination of the antigen, what happens? The macrophage becomes an antigen-presenting cell. And you see clearly here in the figure that the macrophage displays on MHC, which is also HLA, class 2, it displays the non-self peptide. So, by this complex HLA non-self peptide, which cell is going to be activated? Do you remember? Yes, it is the T4 lymphocytes. So it is here where the macrophage moves to the secondary lymphoid organ, like the lymph node, to activate T4 lymphocytes or T helper. So, actually, LB or LT are activated depending on the nature of the antigen. So if the antigen is extracellular, it's the lymphocyte B that is going to be activated. It binds to the antigen through its specific antibody. And you can see here, it becomes a plasma cell which secretes antibodies that can bind to antigens. And when these antibodies bind to different antigens, all of them together form the immune complex. Of course, this activation of B lymphocytes requires also the cooperation with T4 lymphocyte, and this is going to be discussed later in chapter 7. So if the antigen is intracellular, it's the lymphocyte T8 that is going to be activated here. This T8 will recognize the HLA class 1 non-self peptide found on the surface of infected cells, and thus it differentiates into cytotoxic T or T killer that kill the infected cells. Don't forget that both LB and LT8 are activated by T4 or T helper. This is the end of chapter 6, dear life science students. As a homework, please, I want you to do page 132, exercise 2, 133, exercise 3, but please, in exercise 3, I need you to modify the question to analyze the results. What do you conclude? Please also do page 133, exercise 4, and page 134, 135, exercise 8. Thank you, stay safe, and see you soon.